Aging is a multifactorial biological process characterized by the progressive decline in cellular, tissue, and systemic functions. It is shaped by genetic, environmental, and lifestyle factors, leading to increased susceptibility to chronic diseases such as neurodegeneration, cardiovascular disorders, and cancer. Advances in our understanding of aging have identified 14 hallmarks that underlie the aging process, offering valuable intervention points. Peptides, short chains of amino acids, are increasingly recognized as powerful tools to modulate these pathways, offering potential to enhance longevity and health span. The hallmarks of aging provide a framework to understand and address biological aging. Each hallmark contributes to the systemic decline seen with age. Notable hallmarks and associated peptide-based interventions include 1. Genomic instability. DNA damage and mutations accumulate with age. NAD plus precursors help repair DNA and stabilize the genome. 2. Telomere attrition. Telomeres shorten over time, reducing cellular replicative capacity. Epitalon stimulates telomerase to slow this process. 3. Epigenetic alterations. Age-related DNA methylation and histone modification alter gene expression. Chonluten has shown promise in reversing these changes. 4. Loss of proteostasis. Misfolded proteins and aggregates disrupt cellular health. GHK-Cu enhances protein folding and degradation. 5. Deregulated nutrient sensing. Alterations in mTOR, AMPK, and insulin signaling pathways disrupt metabolism. Peptides like CJC-1295 improve nutrient sensing and metabolic health. 6. Mitochondrial dysfunction. Mitochondrial efficiency declines with age. SS31 and MOTSC enhance mitochondrial function and energy production. 7. Cellular senescence. Senescent cells accumulate and secrete inflammatory molecules. FOXO4-DRI selectively eliminates senescent cells. 8. Stem cell exhaustion. The regenerative capacity of stem cells declines with age. TB500 supports stem cell renewal and tissue repair. 9. Altered intercellular communication. Inflammatory signaling increases, disrupting tissue function. LL37 and other peptides modulate inflammatory pathways. 10. Compromised autophagy. Cellular recycling processes become inefficient. MOTSC promotes autophagy to clear damaged components. 11. Dysregulation of RNA processing. Changes in RNA splicing impact protein synthesis. NAD+. Precursors help stabilize RNA processing mechanisms. 12. Microbiome disturbances. Age-related shifts in the gut microbiome increase inflammation and disrupt metabolic health. BPC-157 supports gut lining integrity and microbiome balance. 13. Altered mechanical properties. Changes in tissue stiffness and elasticity reduce functionality. Peptides targeting extracellular matrix proteins hold promise. 14. Chronic inflammation. Persistent inflammation accelerates aging. GHK-Cu and oxytocin exhibit potent anti-inflammatory effects. Peptides act through highly specific mechanisms targeting aging pathways at multiple levels. As research progresses, these peptides may become critical components of personalized longevity strategies. However, large-scale clinical trials are essential to establish safety and efficacy. I'll try my best to keep you abreast of the latest news and research on peptides. Until next time, this is Peter Marino, signing out.